Hello, everyone. We're just getting ready for the start of session 4. As you can see, Adrian Smith is up and live and is ready and raring to go to answer your questions and to head this workshop. As usual, we're just going to give people a little bit of time who are joining. So I'll give a little bit longer because I can see people coming through all the time. So I'll now do, I'll just repeat again for those who've just arrived. So welcome to the U4 virtual conference, Recent Advances in Animal Welfare Science 4. This session, session 4, is a little different from the other ones we've run in that it was due to be a workshop at our physical meeting in Birmingham, and Adrian very kindly and very bravely said he was still willing to give it a go virtually. So he will make a presentation. Please, for this to work, ask questions of things that occur to you. If they're really important, I will try and let Adrian know as he's speaking, but I think in reality, we'll probably have to deal with all the questions at the end. This session lasts for about 25 minutes, so Adrian will speak for about 15 minutes and answer questions for 10. So I am now going to make Adrian the presenter so he can begin the presentation. Thank you, Adrian.
Thank you, Stephen. So you can now see my screen.
I can.
And that was where we finished. So that's where we start now. Are you happy with what's on the screen, Stephen?
Yeah, it looks great. Thank you, Adrian. I'll turn my mic on.
Okay, thanks. This is probably where the hate mail will start coming in, because it's not easy doing a workshop virtually like this. We plan to do it very differently if we'd been in Birmingham. For those of you who just come on, what we're going to do is Stephen's going to collect questions, and we'll do them at the end of my presentation, unless there is something an issue which he feels is so relevant to so many people that he'll butt in and stop me, and I'll take it there. So we have 25 minutes. I've got a lot of slides. Again, I'm going to go through them at breakneck speed because, as I said at the beginning of my last talk, they're all available from the address on the screen now, norwaycorporate.no/U4. So you don't need to write anything down or worry about what's there. I see several.
Yeah,
are you okay?
Yeah, in the background.
Okay, there's some background noise. I think that's somebody with their mic on, but that's been solved now. Okay, I see several hundred of you have seen the prepare video where we talk about the fantastic ability of pilots to fly precisely and reproducibly, and they'll tell you that a lot of the secret of this is using checklists, even on short routine flights, however many times they've flown them before. 10 or 15 checklists for an hour's flight is quite commonplace. And these checklists reduce the risk of them forgetting things, make sure they do them in the right order, and encourage not least cooperation and cross-checking between everybody. And of course, it's too late to read them all when you get to the arrival gate. If the landing gear isn't down by then you probably should have read the checklist earlier. It's also all about what we call threat and error management. All activities we do have some form of weakness or inherent flaw or fault in them, and we have to make sure that we put in layers of defense or redundancy against these weaknesses so that they don't lead to some serious loss. Because Murphy's Law will tell you, and I can confirm, that anything that can go wrong will go wrong, and it will happen when it's least convenient, like when this ventilation fan packed up 1 week before Christmas in Oslo when it was -10 degrees centigrade. And the prepare guidelines draw on a lot of our experience in managing accredited research facilities, where we've tried to think of all the issues involved, both in running the facility and in doing the research there. And I would encourage you to work in the spirit of accreditation, even if your facility is not. And there are a lot of resources out there to tell you how to do that. One of the things that's important is to have a contingency plan, not just against COVID-19, but all the other issues that can and do occur at an animal facility. And we've got some suggestions on our website for those issues that you should be making standard procedures to tackle. And this is particularly important for temporary staff working out of normal hours who don't know the facility as well, maybe don't know where all the solutions to these problems are unless they can read them. And these, of course, need to be revised or supplemented now in the light of COVID-19. And a lot of that is happening. These are some very nice considerations from the British Lab Animal Veterinary Association, and we have a website with a lot of links to similar advice. So it's all about designing, conducting, reporting, and also analysing our animal research to learn from our mistakes. Look for the critical points in the experiment. And if we do this, then we'll find we're on an upward spiral. We're not just going round in a circle, doing things the same way all the time. We're improving. And I don't have time to go into detail now, but there's a lot that we can learn from the accidents that occurred to the space shuttle, where people didn't learn from mistakes or didn't take this threat and error management to its full conclusion. If things do go wrong, then it's a good idea to have somewhere where you can report them. And as you may not have heard of it, I'd like to mention this portal produced by German, Austrian and Swiss lab animal experts, where you can report mistakes anonymously. They're looked at by an expert committee who make an analysis, and you can see here some of the examples of things that have been put there. Really, really nice help, because we all make mistakes, things do go wrong. We also think it's important to have a master plan that it can be really simple on paper like this, where you make a complete overview of the whole of the research, so you don't forget things. They're done at the correct intervals. And you have a contract between the facility and the research group to make sure that there's a correct division of labour and responsibility, to clarify all the stages, and not least make sure the necessary data are recorded. So a researcher doesn't come back to the facility and a year and a half later and say, hey, I think I'm going to get this into Nature, but they want to know the relative humidity in the animal room. What was it? And the facility says, well, I don't know. You said you were going to record it. You see my point. So the PREPARE guidelines, which are a collaboration with Eddie Clutton in the University of Edinburgh, Elliot Lilly at the RSPCA, Christina Hansen at the Norwegian University of Life Sciences in Oslo, and Trond Bratteli, who used to work there and now works at the Western Norway University, were published in 2017 with sponsorship from U4, enabling us to publish it under open access, as an attempt to bring all this together and give advice on how to plan research and testing. PREPARE covers 15 topics, and they've been divided arbitrarily into 3 groups. Formulation of the study and the methods, they're fairly obvious. What's, I think, different from a lot of the reporting guidelines is the bit in the middle, the dialogue between the scientists and the animal facility, how important this is. And there are a lot of issues that aren't typically mentioned in reporting guidelines. I've highlighted those in particular in pink, and a lot of these are also related to the 3 R's. And I'm not going to compare PREPARE with reporting guidelines, but go back to the reporting guidelines you use and see how often the 3 R's are mentioned in them. So PREPARE consists of 2 things. First of all, a checklist where you see all these 15 topics. It's been translated into over 20 languages. There are boxes next to the points on the list, but that's not because this is some mandatory requirement that we're trying to push down the throats of scientists. It's just to, on a voluntary basis, give them things that they should consider and which are probably relevant in their research and to consider when they're at the earliest planning stage from day one and onwards. And as you can see here, there's a lot of references to the 3 R's in this checklist. But the checklist is just the tip of an iceberg, and there is a lot more information on the NORICOPA website. And this part of the Prepare guidelines is growing all the time, because every time somebody publishes a good paper or working party report on something related to animal research, then we add it to the text on the Prepare website. So here's a section on harm benefit assessment, for example, and you can see a link here to a paper about that. So there are links to quality guidelines worldwide on all the important issues. Now, I'm just very, very quickly going to touch on some of the issues that we think about in the animal facility that not all scientists are aware of. For example, the genetics of the animals. If you get mice, even of the same strain from different suppliers, they may have behavioral differences. The diet is so important. We know now a bit more about the effect the microbiota has on animals and their behavior. The contingent suffering caused by things like transport, the stresses in changing housing or social groups or husbandry, and single-housed mice can show symptoms of what we would call depression if they were humans. Then you have methods of handling. We're stopping now picking mice up by the tail, and we're using tunnels and cupping the hands instead to reduce anxiety. NORICOPA was particularly concerned about scruffing, so we made a little video showing how there's actually a better way of scruffing an animal if you have to do that at all. And then there are artifacts caused by poor administration techniques, particularly in small animals. Are you sure that what you injected ended up in the same place every time? Was it painful? Was it even realistic if the organ you're injecting into is small? And in a lot of instances, we don't have a lot of scientific evidence for this. We have to use our own judgment. And I'd like to introduce, for those who haven't heard of it, the 3 S's, who were proposed by Carol Newton just as an aside at a symposium in 1970. She never wrote about them, but they stand apparently for good science, good sense, and good sensibilities. In other words, use your common sense and use your heart as well as your brain. If it would be painful in a human, then give the animal the benefit of the doubt and assume it would be painful to them too. As Carol Newton never published anything on this, Penny Hawkins from the RSPCA and I wrote a paper with our interpretation of the 3 R's, and you'll find a link to it at that address there. The same thing applies to blood sampling techniques, for example. The scientists may feel that it's not a big deal to ask for 0.5 milliliter of plasma from all their mice, but there are a lot of practical issues. Which method are you going to choose? Do they satisfy criteria for good practice? Here's a list of my criteria for good sampling, and only one of the methods at the top of that page satisfies my personal criteria for that. This is the one. It's called Saphenous blood sampling from the hind leg, which we use on a lot of rodents. It was published in this paper, and look at the title. Neither the title nor the abstract said anything about this new method of bleeding animals. And it wasn't until we published that paper that the method really took off internationally. So remember that if you're developing new methods, make sure they get out there where people can see them and where the databases cite them properly, either in the title or the abstract. It doesn't have to be a high impact journal. So there's a lot more on the website about this. We have a lot of links to training films, slideshows. We have a lot of information on educational and training aids that can be used for students and young scientists learning techniques. And as I mentioned before the break, there are a lot of these resources suitable for home learning. What we do is encourage scientists to collaborate with animal carers and technicians from day one. Far too often, a scientist will go to the head of facility, have a little one-to-one meeting in the office and say, oh, I think we found the cure for Alzheimer's. This is what we plan to do, but without involving the technicians. And that's a big mistake, because they have a right to know. They'll be more motivated. They, more than anybody, know the possibilities and the limitations of the animal facility. They have a large range of practical skills, and they may not have done this on rats before, but maybe they did it on guinea pigs last year and can adapt that technique to your experiment. They know the animals best and the animals know them best. So the animals will be most calm and harmonized if the technicians do the work. And if we don't involve them, we just create this anxiety, depression, opposition to animal research we don't want and limit their creativity. Back to the shuttle tragedies I talked about, we need a culture of care because a lot of the things that went wrong with those 2 shuttles were related to this. I don't have time to go into the details now. You can read about it on the net. But there were weaknesses which people knew about. There was pressure to launch. There were bad management decisions. And a lot of these seemed fairly small, but when they occur simultaneously, they cause disasters. And if you don't believe small details are important, try spending a night in a tent with a mosquito. As I said before the break, we also host the website of the International Cultural Care Network, which is all about a demonstrable commitment to these 4 points here, beyond compliance with the law. And there's a lot of good resources there about how we can improve one health, one welfare, as we've heard earlier on this meeting. This is an infographic made by Penny Hawkins at the RSPCA, used by the Cultural Care Network. I also said that we've recently launched a refinement wiki about the same time as the COVID-19 pandemic started. So work on this hasn't really taken off yet, but we would really invite you to contribute to that so that small refinements that aren't yet published can be put out there quickly to get to people's attention. We also produce a lot of guidance ourselves. Here are some of them on relevant issues. We also have a website with recordings of, rather the PDF files of all our guest lecturers at Noricobus meetings. We have a very comprehensive webinar and meetings calendar, which we're updating all the time with worldwide meetings. We produce a newsletter 7 or 8 times a year in English, which you're welcome to subscribe to. You can do that by a link at the bottom of every page on the Noricobus website. And to summarize, to give you time now to discuss this, I'd just like to say that PREPARE is not just another set of guidelines. In fact, it's not just another set of guidelines, because it's, in addition to the checklist, this big website where we're putting resources that people like you have developed all the time. We're adding to this all the time to increase our knowledge base on how to do good research, whether it be with animals or not. So we hope that this will help scientists, when they go to a journal with a manuscript, sail through the review process, because they would have thought and attended to all the issues which the reviewers and the editors are concerned about. Bringing us better science, improved animal welfare, advancing the 3 R's, and giving us a safer working environment. So I'd like to end up by thanking our main sponsors, not least EUFOR, who have financed work we've been doing for many years now, and remind you that all these slides were available at noricobus.no/EUFOR. So that's all I was going to say. Now we can.
Okay, thank you, Adrian. Yeah, we've got about 6 or 7 minutes for questions. So,
yeah.
First one from Benjamin Lacour says, "Great topic to talk about. I wonder how this topic is brought to more hardcore scientists. Fields such as neuroscience and pharmacology, just to cite a few, are heavy in animal use, especially procedures that are invasive or likely to induce suffering. Are they receptive involved? If not, can you elaborate on how to improve this?"
Yeah, they're certainly receptive, partly because if you go to a meeting site, like say the International Transgenic Society meeting, which I was at, was it last year, it's a small portion of the Congress, and it looks a bit exotic in a way. It's not a typical nerdy lecture on CRISPR techniques. And if you have to be, of course, a bit diplomatic and and say, you know, this will actually improve your science and we're not trying to stuff it down your throats, then they normally accept it very well. And I think the lab animal community has to be far better at going to those meetings, the pharmacology meetings, the tox meetings, the transgenic meetings, rather than just preaching to the converted.
Thank you. Kathy Dwyer asks a question. Interested in the idea of the review process when things go wrong, which seems very important. How would you advise this is done, as it could be emotive or confrontational?
Sorry, was the question, how is it done?
How would you advise this is done? Yes, to do with the review process when things go wrong.
Right. Well, I mentioned specifically that portal that the Germans have produced, CS Lust, and you fill out an anonymous report there, and then it's looked at by an expert committee who give their opinion of the situation. I don't know it in detail because I've never submitted a report to it myself, but I can talk afterwards with the main person behind it and ask her to give me more information. We have a bit of a description of the portal on the Nora Rokocpa website. So if you search for CERSLAS on our site, you'll find that information. And there is nothing against applying that principle locally as well. If you can get the right people together, then you could have some means of discussing this sort of thing on a local level without getting people's backs up. If you have a good culture of care at the facility, then it's a lot easier to discuss the things that went wrong. And this is the whole point of why we promote a culture of care, because it means even the lowliest animal care or cage washer can have the confidence to go to the boss or their manager and say, look, you know, I really think we should be doing things differently. It's all about a culture of challenge as well as a culture of care. So if you have a good climate at your facility, then it will be a lot easier to discuss mistakes. And the EU directive says very specifically in the recitals before the main text that we should foster this climate of care at a facility.
Okay. Question from Hugh Gollidge, and it might have to be the last one looking at the time we've got left. What do you think about study pre-registration and even publication of peer-reviewed registered reports and study plans as a way to encourage people to prepare? This way we could check that the guidelines have been followed before the study starts.
Yeah, it sounds a really good idea. The one thing I'm concerned about is that prepare will be given a sort of mandatory status in scientists' eyes, that they will feel this is just another loop they have to jump through. We're trying as far as possible to do this on a voluntary basis, that people open their eyes for the fact that it's too late to discuss all the details when you get to the reporting stage. Whether this should be done exclusively through pre-registration or other ways is something we should carry on discussing, but in principle, in my opinion, it sounds an excellent way of doing it. But
okay.
I do want this to be voluntary and that people understand that we get better science as a result.
We've got probably time for one last question. A lot of this talk was aimed at animal facilities. Presumably, Mandy Lucas asks, prepare transcribes for field trials for farm research into animal welfare science.
Absolutely. Our aim was actually to, by putting the checklists available on the net so easily, was to let, say, fish folk or wildlife experts adapt it to their own needs. And certainly when I'm talking to them, I do phrase things rather differently and give other examples. But I work closely with those researchers. We have a lot of wildlife and fish researchers in Norway, and they understand the principles. We just need to give them examples of other animals rather than mice and rats.
Excellent. Okay, well, thank you. It's almost bang on time. So well done, Adrian. A really interesting presentation. And I'm going to have to stop it because we've got to launch session 5.2, which I hope everyone who's here at the moment will join as well. So thank you very much, Adrian, for a really interesting presentation. As always, if you have any questions, then please, you can also email Adrian directly, and I'm sure he'll be very happy to go into more detail.
Would indeed.
So thank you. Thank you very much.
Bye.